This protocol uses a simple method for enhancing the reliability of a 3D cell culture model without the use of any special equipment. The main advantage of this technique is that we can conduct repeatable 3D culture experiment by controlling the initial culture condition within the cubic device.
After preparing a 5x5mm polycarbonate cubic frame, place the frame on a pre-cooled slide in an ice box and use a pipette to add 12 microliters of preheated 1.5% agarose from the top face of the cubic frame to the bottom surface. Spread the agarose to obtain a flat surface and allow the polymer to cure. Use tweezers to slide the frame to the edge of the glass and rotate the frame so that the open side is facing down before placing the frame back onto the slide. After filling the next two surfaces of the frame with more agarose as just demonstrated, drop the agarose into a container from an open face to form an agarose wall on the fourth and fifth sides. To set up an initial cell cluster shape, inject an appropriate extracellular matrix for the cell culture of interest into the hybrid gel cube culture space and set a fabricated micromold onto the cube. Then place the cube in a carbon dioxide incubator for 25 minutes at 37 degrees Celsius. When the extracellular matrix is cured, carefully lift out the micromold so that the matrix does not deteriorate. A pocket in the desired mold shape will be fabricated in the extracellular matrix. To load the cells, concentrate the experimental cell suspension after centrifugation in the appropriate experimental cell culture medium, and inject the cells into the pocket.